Good afternoon. My name is Sherlyn Contreras, and this summer I conducted research in Dr. Maureen Hansen's lab under the mentorship of Dr. Savan Bentolila. Our research focused on the functional characterization of ORRM encoding genes in Arabidopsis thaliana. To begin, RNA editing is a common type of RNA processing that occurs in many species, such as viruses, trypanosomes, squid, plants, and us, humans. Specifically, RNA editing is a post-transcriptional process that converts cytosines to uracils in both the mitochondrial and plastids mRNAs. It occurs 37 times in the chloroplast, but 619 times in the mitochondria. Now, in this image below, it shows the structural change that occurs during this editing process. The red circles emphasize a cytidine deaminase reaction. Now, why is a C to U editing needed? Well, editing corrects organellar transcripts, meaning that most known editing events result in the restoration of a codon, either for a conserved amino acid or a stop or start codon. And the latter is shown in the example here. In the chloroplast, in the Arabidopsis chloroplast and the HT transcript, the cytosine in the first codon is edited to uracil. Now the AUG translation initiation codon is restored. Now that we have explained why C2U editing is needed, let's see what it looks like when it's lost. The loss of editing can result in various phenotypes. In the example of this particular C target, chloroplast ACCD794, there is no real difference of phenotype between the wild type and the mutant. However, in some cases, the mutant can show a pale yellow phenotype. In more severe cases, morphological defects can be seen in the mutant. And in this case, that was dwarfism. In the most severe cases, the loss of editing at particular C targets could lead to the plant's death. So I've explained why C2U editing is needed and what happens when it's not there. But the question about specificity still lingers. Or in other words, how is the proper cytosine selected for this editing process? The proper cytosine is selected for editing by a combination of cis elements and trans factors. The cis element is labeled in red as a recognition sequence. Cis element sequences vary for different C targets. Additionally, cis elements are recognized by a particular protein. In this example, the star is a C target, CCMFC333, and it's being recognized by the PPR protein. The edosome complex also has RIP and ORRM in it. On this topic, plant RNA editing requires a plant edosome made up of multiple protein complexes. The PPR is an RNA binding protein whose role in the edosome is to bind upstream of that specific target and hence control the specificity of the RNA modification. Going back to the figure, we see that there's PPR and again, RIP and ORRM. ORRM standing for an organelle RNA recognition mode of containing protein factor. While the roles of editing for ORRM and RIP, both of them being protein families, are not as clear as a PPR, they are just as essential to the edosome. ORRM1 is known to be part of a clade where most proteins are organelle targeted. 5 additional ORRM editing factors highlighted here have been characterized. The top ones have been localized to be in the plastid and the bottom ones in the mitochondrial. As you see here in the black bracket, these are 5 uncharacterized ORRM genes. However, not all of these take part in editing, such as the one highlighted in blue. RPS19 is actually a ribosomal protein. tDNA likely disrupts the gene and its function. So we hypothesize that the insertion of tDNA in the genes encoding the uncharacterized ORRM proteins might impair their function. If an ORRM protein is important for RNA editing, the mutation will decrease editing. Importantly, the mutation may also result in morphological defects, which have been observed in other mutant plants affected in plant organelle editing. In our research, we focused on 11 different tDNA populations. All were pulled from the 5 uncharacterized ORRM genes, which is seen and labeled in the gene ID column. Localization was also annotated in this table when available. In these columns, P is for plastid and M stands for mitochondrial. And you can see these letters here. Now, in the example of the first tDNA population, it was localized to be in the mitochondrial through MESPEC and also through the 2 predictor localization programs, predictor and target P. Once more, the first group ends with a C. The tDNA population names end with a C when they're supposed to be homozygous populations. However, production annotations can be wrong, so this calls for the need to genotype and sequence these plants. For each tDNA population, 24 plants were grown in chambers. 6 out of the 11 groups are pictured here, and they were about 1 month old when this picture was taken. The tDNA insertions obtained from the stock center are either homozygous or segregating. The first step is to genotype these populations in order to conform the homozygosity of the mutants or to identify such mutants. I began this process with DNA extraction. First, I collected 2 to 3 small leaves and placed them in a tube. Then I added 100 microliters of the extraction buffer. The leaf tissue was drilled using a power drill, and 400 more microliters of that buffer was added. Later, this tube was incubated in a water bath of 65 degrees Celsius for about an hour. After 500 microliters of the chloroform isoamyl alcohol was added. Then I took this tube forcefully between 2 tube racks and centrifuged it. This allowed for 2 layers to be visible, the aqueous layer and the organic layer. And the aqueous layer is where the DNA is. So I went ahead and transferred that aqueous layer into a new tube. It was first washed with isopropanol. And in the circle, there is the DNA that's being precipitated via the wash and washed again with 70% ethanol. And once again, you can see the DNA is precipitated. I poured out the excess liquid in the tube and suspended the DNA pellet in distilled H2O with RNase. After extractions were done, I continued with PCR amplification. I added 1 microliter of the DNA sample to 2 wells. Keep in mind that the DNA samples is coming from the same plant. So 1 well was incubated with the mutant solution, and the other was incubated with the wild type solution. The difference between these solutions is that the wild type has the left primer and the mutant has a tDNA primer. Then it was placed to the PCR cycles, and I visualized my PCR results using gel electrophoresis. To further explain, the left and right primer in the wild type mixture are an appropriate distance apart to still make it possible for the PCR to amplify this gene. However, when the mutants have this tDNA insertion, now the left and right primers are too far apart. And that's why the mutant solutions, instead of having the left primer, it had the tDNA primer. As explained before, the 11 groups of tDNA can either be homozygous or segregating. This is a PCR result of a homozygous tDNA population. When treated with the wild type or incubated with the wild type solution, no bands were visible. Only, as seen here, only the ladder and the controls were visible. This means that no wild type alleles were detected in these samples. On the other hand, when the samples were incubated with the mutant solution, we can see that there's multiple bands visible. Therefore, multiple mutant alleles were detected. But in the cases such as 5, no band appeared. This simply could be due to not having enough DNA from the extractions. So even though not all the bands appeared, we could still annotate this to be a homozygous mutant population. The other genotype option for our tDNA groups is for them to be a segregating population, which means that it shows more than just one genotype option. This is a PCR result for a total segregating population. For well 3, and keep in mind that well 3 both hold DNA coming from the same plant. Only when incubated with the mutant solution did a band appear. So therefore, this individual plant only has the mutant allele, making it a homozygous mutant. In the case of 12, only a wild type allele was detected, therefore making it a homozygous wild type. Lastly, in cases such as 17, where both the wild type and the mutant alleles were detected, this individual was a heterozygous plant. This table shows our genotype results for 9 of the tDNA populations. Under the 3 different genotypes, there's an X if this genotype was present in that group. The yellow highlighted shows single genotype populations, green shows single genotype populations, while the yellow is segregating populations. Now, marked with 2 red arrows are 2 misannotated tDNA populations. As explained earlier, the C in the end of the tDNA population name marks these groups to be a homozygous population, but this was actually not the case. I found these 2 groups to actually be misannotated and to actually be segregating populations and not homozygous. After all groups are genotyped, the homozygous mutant individuals were prepped and sent for Sanger sequencing to localize the tDNA insertion. In the example of cell tDNA, of this cell tDNA population, we found that the insertion was actually misannotated to be in the first intron. Through our sequencing, we localized it to be at the end of the first exon. Our research will be continued by performing an RNA analysis. I've already begun with the RNA extractions, but the next step is to perform a reverse transcription PCR. This process will amplify the organelle's cDNAs and their transcripts. Then RNA sequencing will be used to quantify the RNA editing at these C targets. This means that we can see if there's a decrease in CDU editing. And finally, will help us answer the question if editing was affected by mutation. Finally, I would like to thank NSF and Cornell University for supporting my research and internship here. I want to thank the wonderful people in my lab, and especially Dr. Pinto Lila for his great mentorship and kindness. I want to thank you guys so much for listening. If you have any lingering questions, please don't hesitate to reach out to me. This email.